Uh, all right, thanks everybody for coming. I am going to share or do a quick tutorial on how to register, or one way, because there are several ways, but one way to register your data to an atlas. And once you've done that, how to move objects back and forth between your subject coordinate system and your atlas coordinate system. And I'm going to try to do this very quickly because we're starting the meeting 35 minutes late today to accommodate a previous meeting. So I'm going to ask, this is not really in the spirit of how to, but out of necessity, I'm going to ask that nobody ask any questions until after 2 p.m. That way I can try to get through all of the content I want to present. And then I'm happy to stay as long as people want after to answer questions at that point. I'm going to
Oh, Greg, we can't hear you.
do a few slides, then I'm going to get into just a really quick view of what this whole problem is about. What is an atlas? It's usually an average representation of something, typically anatomy. Classically, an atlas was something published on paper in a book, and it might have tons of histological sections present with different types of stains that would tell readers this part of the brain, if it's a brain, this part of the brain relative to this other part of the brain is a particular white matter tract or a particular nucleus of a given cell type or something. And it was meant to orient readers to how different components came together to make up the piece of anatomy. And I say it's usually an average representation because it was usually one or a few subjects that were supposed to be representative of an entire population. Like this is the wild type lab mouse, hippocampus, for example. Typically within an atlas, there's lots of data attached to it. So there could be annotations. So all the examples I'm going to give are from zebrafish because that's my field and that's the example that I'm going to use for doing the registration is zebrafish data. And you can see when I toggle the slide, we go from just a picture of the zebrafish brain to the same picture with a bunch of manual annotations plotted on top of it. And so this is where an expert anatomist went in and drew labels all throughout the brain to indicate this is a particular population of cells, or this is a particular brain region, so that in your digital image, you can find those regions of interest. It might also contain many different channels or expression under specific, which is expression of specific genes under specific promoters. So you might have your average image of the brain, but then you might also have hundreds of other images that visualize only specific structures, which give you even more information about where those structures are located, what they project to, what type of genes they express, things like that. So generally, an atlas contains lots and lots of useful data that might help you in your experiments interpret your own data. And then all of those forms of data are typically, in a modern digital atlas, represented with high quality images that you can use to register your own data to the atlas, explore the space of images yourself online, things like that. And all of these examples are pulled from this atlas called the Z-Brain, which is of a 6 days post-fertilization zebrafish. And I'll open this really quick and we'll take a look at the website for the Z-Brain just to show more examples of what I'm talking about. So when you open this website, you get this image right here, which is the most commonly used image from the atlas, but there are tons and tons and tons of other images present in the atlas. So you see, I've just switched to I picked it at random, some Gal4 line that's only expressing fluorescent reporter in a subset of cells that are positive for a specific gene. And there are hundreds of these in this atlas. And in addition to that, there are also, in the download section, all of these other data sets associated with the atlas, like the manually generated labels that tell you where these brain regions are. So if you're doing experiments using whole or even partial zebrafish brain, using the Z-Brain or one of the other interesting zebrafish atlases can help you import tons of information about zebrafish brain areas into your own experiment to help you interpret your results, generate hypotheses for future experiments, confirm or validate specific observations you've made with respect to other fish, and things like that. Okay, so that's what an atlas is. What does it mean to register your data to an atlas? It means that for every coordinate in your data, for every I, J, K pixel location, you want to know the corresponding coordinate in the atlas data. So you pick a random location in your data, you want to know what the corresponding point is in the atlas data so that you have a complete mapping of every location in your sample to every location in the atlas sample or in the atlas data. The mapping object that tells you this correspondence is called a transform, and you get transforms from registration algorithms. And once you have this transform, you can start moving stuff back and forth between the two spaces. You can take manually annotated labels from the atlas and move them onto your image to interpret your data. You can take your data and move it into the atlas space to contribute it to the atlas so future users can see it. Or you can do this for, say, 100 of your subjects, register all of them to the atlas, and now they're all in a common space where you can average your observations between your different subjects and know that you're averaging the same brain areas together. Okay, last slide before I get to actually looking at code. What does it mean to move objects from an atlas to sample or from sample to atlas? This is just reiterating a point from the previous slide. Moving data from atlas space to individual space, like annotations or channels, this is an example of doing that here. So here's the atlas data with the manual annotations over it again. And then here's an individual subject, not part of the atlas, but who is registered to the atlas. And I've taken all of these manual annotations and I have used the transform to put them on top of my individual subject. So now in my individual subject, I can say, oh, this is the habenula, or this is the neuropil of the optic tectum, or any other brain region that's present in the manual labels. Okay, so let's look at the code that I use to actually do this. I'll pull the Jupyter notebook up in a second. All this stuff that's open are images I'm going to reference during the tutorial to show you the outcome of the individual steps of this process. Okay, so here's a Jupyter notebook. I'm using a library of image registration tools called BigStream. I wrote it myself, but it leverages a lot of really great libraries for doing image registration like Simple ITK and Dask. But there are other tools that you can use to do image registration like Ants, Greedy, John Bogovic's great Big Warp package, lots of things. And you can also combine a lot of these tools together, since a lot of the time, some of them will have a feature that's really important to you. And like you might need two registration tools in combination to really complete your job. And all of them are compatible with a little bit of knowledge about how they can work together. All right, so this first cell, I'm just reading in my input data. I'm reading in two different images. One I'm calling fix, and one is a channel from the ZBrain Atlas. That's these two images here in the upper left-hand corner of my screen. So fix is a confocal image of a whole 6 days post-fertilization zebrafish brain. It was collected by someone who is here. Emmanuel Marquez Legarreta collected this image, so thanks for that. And it's, like I said, one image from a single zebrafish. And then this other image, the ZBrain eLavel H2B-RFP, is one channel from the ZBrain Atlas. And this is not a single fish. This is actually many different images from different fish, all registered together and averaged. Remember, I said an atlas is usually an average representation of something. And so this is a combination of many different subjects' data to produce something that is not idiosyncratic to the individual variability of one sample. It is representative of the population of 6 days post-fertilization zebrafish brains. So I'm just going to load those two things into memory. Oops. Well, first I'm going to get rid of a typo that I just made. Now I'm going to load those things into memory. And notice I'm also defining here just explicitly what the voxel spacing is. So these, of course, are digital images. They're made up of individual voxels. And each of those voxels corresponds to a physical size in micrometers. And those sizes are different between the two different data sets. And any registration algorithm should, at some point, be asking you for what these values are. And if it's not asking you that, it's probably not doing things correctly. So it's very important that you know these values for both the atlas data and your individual data. Okay, so that data is now in memory. The next thing that I do here, which is not a requirement for registration, but can be very helpful, is I'm going to mask these two data sets. I'm going to generate in an automated way a foreground mask that tells me which part of the rectangle that the image makes up contains brain and which part contains non-brain, just background. This cell takes a little while to run. So to speed things up a little bit, I already have these results computed, and this is what they look like. And really quickly, I'm going to superimpose them on top of the images that they correspond to so we can visually inspect that they actually look good. So here's a copy of the fixed image. And here's a copy of the Atlas channel. And now I'm going to use Fiji to merge these things. I'm going to put my fixed mask in the red channel and the copy of my fixed image in the gray channel. It keeps sending them to my other desktop, so that's why I'm dragging Windows over. So it's not a perfect mask, it's automatically generated by a pretty basic algorithm, but all it really needs to be is roughly correct. It's going to help the registration algorithm ignore a lot of work that we don't want it to do. We don't want the registration algorithm to be paying attention to background voxels at all. So this looks pretty good. Now let's take a look at the mask for the Atlas channel. Move mask. Gray is the duplicate of the mask of the Atlas channel. And here they are superimposed. So again, we've got pretty much what we want, a very rough estimation of foreground versus background. It may not look like it, but at least for the zebrafish brain, this foreground background mask it allows us to ignore almost 60% of the volume of the entire image. So if we can tell our registration algorithm that at 60% of our voxels aren't important for the computation, it can speed things up considerably and also give you more accurate results, because then things going on in the background like artifacts or noise are far less likely to affect your final outcome. Okay, so that's the masking. It's still running in the Jupyter Notebook. Like I said, this takes maybe like 8 or 9 minutes. I'm using another function from BigStream to do this foreground background masking. It's called the level set function. And I'm doing a little bit of extra corrections on the masks that it produces. And so this takes about, between 5 and 10 minutes to complete. So while that's running, I'm going to move on to actually start talking about the alignment. So like I said, I'm using my BigStream package to run an image registration between these two images, and I'm using a function called the alignment pipeline. The alignment pipeline is more than just a single registration. It's a way to build a customized pipeline to register two data sets. And you can do that in a very, very simple way, like just running one single step, or a very, very complicated way, chaining many different steps together of different types. In this case, we're going to chain together two steps. We're going to do something called an affine registration and a deformable registration. More or less, the affine registration is going to put the two data sets roughly on top of each other and make sure they're the same size. And then the deformable registration is going to move individual edges and local regions to make sure that those are matched with each other. Each of those two steps themselves has a lot of things that you can configure in order to influence how aggressive or gentle the registration is in manipulating the data. Most of these values are in pretty standard ranges, and the algorithm's pretty robust to the choices that you make for them. But I like to keep them exposed so that the user knows that they're available and that they can start tweaking them and messing around with them in case the results they get don't look great. They have a lot of options to dig into to start trying to change things around a bit. So like I said, we're going to run an affine alignment, and these are its parameters, a deformable alignment, and these are its parameters. The two masks that we made are only being used for the deformable step. Notice they're not arguments in this affine step. Those two steps are bundled together into a recipe, if you'd like, which the alignment pipeline takes as input right here, this steps argument. And you can see how this can be this steps recipe is completely arbitrary. It can have 2 steps like this, or it can have 1 step if you just want to do a single alignment, or it can have 100 steps. It's completely arbitrary, and they can be in any order. You can do an affine and then a deformable, and then 2 more affines, and then a rigid alignment, and then there's I think 6 different types of alignments available in BigStream. And so any combination or order of those 6 is permissible here. The other arguments that the alignment pipeline is going to take are the fixed and moving images that we've looked at here, fixed and moving, and then the voxel spacings that we defined in the first cell. And then this last keyword argument, return format, because we're doing 2 different steps, we are going to solve for 2 different transforms. The alignment pipeline can combine those for you if you like and return a single object, or it can return separate objects for each of the transforms that you solve for. And that's what I'm telling it to do here. Please give me a separate affine object and a separate deformable object. Finally, you want to know if your alignment actually worked or not. Once you run this cell, it's going to start. Let's see if the masking finished yet. No, the masking is still running. It'll finish in a couple of minutes, and then this align cell will start running, and it'll start spitting out a bunch of output that we'll be able to talk about. But you never really just want to trust the text output of an algorithm. You want to visually inspect what it did to your data. The algorithm might tell you, hey, I succeeded, I did a great job. The metrics were optimized. Good work. But you have to visually confirm that. And so now that we have these transforms, our affine and deformable transform, we want to use them to actually resample our moving image data and check to see that it now visually looks similar to our fixed image data. And that's what this function apply transform is doing. It takes almost the same arguments as the alignment pipeline, the fixed image, the moving image, and the voxel spacings. And it also takes this transform list, which is an arbitrarily long list of transform objects. These 2, here it's taking the 2 transforms that we got as output from the alignment pipeline, but it can take 100 transforms if you want, and they don't all have to come from BigStream. A pretty common workflow for me is to run some automated alignments using BigStream's alignment pipeline, see that there are a few areas that need to be corrected, and then use a different tool to correct those areas, one that's more suited for it. For example, Big Warp. And then bringing all of the transforms from all the different tools that I've used, BigStream, Big Warp, Greedy, Ants, whatever I've used, back into BigStream, and then using this apply transform function to compose all those transforms together and actually resample my moving image data. So that's all that this cell is doing. The final chunk of code here is just actually storing some of these results on disk. And now that the alignment has actually started, I can talk about what some of this stuff is that's getting printed out. So this alignment is an iterative algorithm. It makes a guess, and then it checks how good is my alignment, and it gives it a score. And then it makes a better guess, and it gives that a score, and a better guess, and it gives that a score. And each one of these lines is one of those guesses. And the metric value is that score for how well aligned the data is. And you want to see that this value is smoothly and gradually increasing in negative magnitude, or I guess you could say decreasing, getting more negative over time. And we see that it is, which is great. And you also see that it's spitting these iterations out quite fast. And that's because I've configured the alignment to be very quick, but still quite accurate. The choice of parameters that I'm using here is quite fast, but still accurate. And now it says registration succeeded. That means it has completed the first of the alignment steps. The affine part is done. Now it's going to do some thinking, and in a minute, it'll start the deformable steps. And while it's thinking about that, I'm going to let's look at the final result of just the affine part. I have it open separate from the just the deformable part, which I have to move in a weird way because the zoom window is blocking where I want to grab the image window. There we go. Okay, so this image here, aligned affine only, is the result of only the affine part of the image registration. So I'm going to duplicate my fixed image. I have a second copy, and now I'm going to put the aligned affine only image on top of it. So we're coming up on 2 p.m. And I would say I have probably no more than 10 minutes left of actual presentation, just for those of you who have something else to do or who need to go. I know I'm a little behind. It's hard to squeeze something into 25 minutes, but I will try to go fast. Okay, and then here's affine aligned only. Okay, so I'll make it bigger so we can take a look. So like I said, the affine step just puts the two data sets roughly on top of each other and makes sure that they're approximately the same size. That seems to have been accomplished here. The correct structures are roughly in the same location, and the fish brain overall is roughly the same size. But of course, there are still really large areas that are not correctly registered, like this boundary. And you can see that along the z-axis, this habenula structure is not quite located on top of the right thing. The forebrain probably needs to tilt down a little bit more in order to be correct. But an affine registration alone is not going to be sufficient to capture those types of transformations. So that's what the deformable step is going to do. So the deformable step has begun running, and similar to the affine step, it's going to print out a bunch of these lines. And you still want to be checking that the metric that is comparing the images is gradually decreasing in, or becoming a larger negative number, and that seems to be happening. This will take probably another 10 to 15 minutes, so we're obviously not going to wait for it to completely finish, but we are going to look at the result, which keeps hiding behind that little drop down from Zoom, so I can't move the window. All right, here it is. This is aligned. This is the result of applying both the affine alignment and the deformable alignment. So now I'm going to put this on top of the fixed image data, and we'll see if we were able to correct those areas in the affine only alignment that were still incorrect. Okay, red fixed image, green aligned image. And here's the result. So we're looking considerably better. So like this area in the optic tectum, which is where the most obvious error was before, is pretty well corrected. And this area in the habenula is more correct. I wouldn't say it's perfect, but it's pretty close. Another thing to bring up with atlas to subject registration is expectations. You can never expect an atlas to subject registration to be perfect at, say, the single cell level accuracy. Two brains are never the same at single cell level. The exact location, there might be a different number of cells in the two brains, and even within a specific brain region, there might be a different number of cells. And so the best that you can do is usually accuracy up to something like maybe 20 to 30 microns is like a reasonable spatial scale to say that two things are accurately aligned in an atlas to subject alignment. If you are asking yourself, are the right cells right on top of each other, then you're asking a question that doesn't really make sense for an atlas to subject alignment problem. So I would say that this alignment is pretty darn good, especially for the speed and ease at which it's able to be computed in Python, but it's not perfect. And the area that I like the least is down here. This is a place where automated alignments really struggle, where you have these really thin, long structures. They're hard for the automated alignment algorithms to grab onto and move in a way that doesn't tear them or shred them. So the reason I didn't try to correct this in an automated way with a more elaborate automated algorithm is that I think a better approach here would be to use Big Warp. You can take this resampled aligned image and open it in a manual alignment tool like Big Warp and correct this yourself with just a few minutes of clicking. You can then export that transform from Big Warp, and you have a transform stack that gives you sort of the best of both worlds. A good automated alignment that took care of a large percentage of the data for you, and then the assurance that even areas that are hard to align in an automated way are still very accurately aligned because you went in and manually corrected them. And with a little bit of investigating, it's perfect. The transforms from BigStream and from Big Warp, and really from any other alignment tool for that matter, if you know what you're doing, are all compatible with each other and can be combined in something like this transform list, or even in an ImageLib function that composes transforms. They're all, with a little bit of effort, compatible with each other. So, all right, that's the entire registration part of things. The last part of this presentation, which is actually, I think, the most important because it's the least talked about. I find I'm always teaching people how to register things. And then I'm always like, okay, great, we're done. And then they're like, but hey, what I actually want is these labels from the atlas on top of my subject, and I don't know what to do next. So it's this part of moving objects back and forth that is often not discussed. And so that's what I very briefly want to talk about. Okay, so we have a good alignment. Now, how do I move objects from atlas space to my subject? And actually, before we look at this slide, let's look at the cell where I actually do this. And it's actually quite simple. It's not different from moving the atlas channel data to the fixed channel. It's basically the same thing. In this case, our labels are represented as a multi-integer image. Let me show you in Nipari. I really like the way Nipari shows labels, multi-label images. So here's our atlas channel with all of our manual annotations on top of it. And this multi-label, the labels part of it, just this part of it, is an image, just like the actual atlas channel data is an image. And so we can warp it in the same way that we warped the atlas data. We can use, for example, the BigStream apply transform function. So all I'm doing in this cell is reading in that multi-label image representation of the manual annotations from the atlas. And then I'm calling the apply transform function, and I'm giving it the fixed image as a reference so that we know how to resample the labels data themselves, and then the two voxel spacings, just like before, and the transform list, just like before. Now, the only thing that's present now that wasn't present before is this interpolate with NN, which stands for nearest neighbors. And that's because of the data type. The data that we're moving now is strictly integer valued. And values 4 and 5 mean very distinct things. 4 might label the subiculum or some random part of the brain, and 5 might label some other part of the, I'm not a neuroscientist, so I don't know, some other random part of the brain. And those two parts of the brain might be very far apart from each other. So an intermediate value between them, like 4.5, is a totally meaningless number in the context of that image. So interpolate with nearest neighbors says, when you are warping the multi-label image data, you should never be producing values that are non-integers. You should only ever be in the output image producing values that are integers. So that's it for moving the labels data from the atlas to the subject. And if you do that, you can get something that looks like this. So here's our fixed image. It's the same as the one that's open in Fiji. This one here, I just have it open in Nipari now because I really like the multicolor maps for multi-label images in Nipari. And you can see that now the labels are present on my individual subject. And this enables you to do all kinds of interesting automated analysis on your individual subject. Maybe you've collected functional imaging data from that subject, genetic data from that subject, projectomics, transcriptomics, proteomics. Maybe you've done antibody staining. Maybe you have Gal4 lines. Who knows? But in your individual subject, you have some observation, and you want to combine that observation with the anatomical regions from the atlas. And now you can do that in a computational way. Okay, last thing to say is, what if I want to move things from the subject to the atlas? All I've shown you so far is warping data from the atlas to the subject. And so I'm just going to put this slide up really quickly so that people can either screenshot or look at in the recording, which is a summary of how to move the things you want to move. So on the left, the rows here are, say I want to move something from the moving image to the fixed image. In this case, the atlas was the moving image. Or I want to move something from the fixed image to the moving image. So if you're taking something from moving to fixed, you use the transforms that the registration algorithm just gave you. And that's all I've shown you examples of so far in the Jupyter Notebook. If you want to move something from the fixed image to the moving image, you're going the other direction. So you want to invert those transforms, and those are the objects that you're going to use to transform your data. So the last cell in this notebook is me showing you how to invert transforms, at least using BigStream. So BigStream has a function called invert displacement vector field. And here, these two lines is where I'm inverting the two transforms that our image registration gave us. Inverting the affine transform is not actually a BigStream function. It's really simple to do. It's just a matrix. So I'm just calling a matrix inversion function from NumPy. It would be silly to wrap that in BigStream and claim that it was some fancy thing. So you just can call NumPy linear algebra invert the affine matrix. And then the deformable function, this is a little more complicated to do. So BigStream does it for you. Invert displacement vector field. It only takes the deform itself that you want to invert, and then the spacing of that. There are some optional arguments too, to either increase the accuracy of this algorithm or make it run faster, but the defaults are usually suitable. And then I save those two inverses. And then I'm calling again the apply transform function, but notice that I'm now have reversed the order of everything. Instead of fixed move here, I have move fixed. And instead of fixed spacing, move spacing, I have move spacing, fixed spacing, because now I'm moving things in the opposite direction. So everything's playing sort of the opposite role that it was playing before. And then the other thing often overlooked here is the transform list. In the transform list, I'm using the inverse version of these transforms, but I've also reversed their order. Just like reversing the order of the images, I've reversed the order of the transforms as well. And that when you are going the opposite way, you have to do that. There's another interesting point about moving geometry data, which is another common thing that people want to do when they have two registered data sets. Geometry data is anything that's not like pixels. It's usually something where you have like an explicit coordinate value, like I found a dot of interest at location XYZ, or maybe I found 100,000 dots of interest at the following XYZ coordinates. When you move objects like that, you have to use the opposite conventions. And I definitely don't have enough time to explain why. It's notoriously difficult to explain. John has good slides explaining how it works, and I'm happy to explain it to folks later. But when you are moving geometry data, like coordinates or meshes, surfaces, extracted, triangulations, things like that, that's also geometry data, then you have to use the opposite conventions as what you do for image data. And that about wraps it up. That's the whole thing. So I'll stop now. And if anybody has questions about any part of this, please let me know.
So, Greg, I have a bunch of questions.
Sure.
So for these images are very nice. There's no noise. They're very complex. As a person does registration, when do you decide when you can't register something because it doesn't have enough complexity or structure to it?
That's a great question. You get a feel. I mean, I have a good feel for that because I've done so many registrations throughout the years. And so like my personal answer is I'll try anything. Is my first answer is like any data set, I'll be like, well, we can always try it. And then I'll usually look at the output and determine if the alignment failed, how did it fail? And does it look like a type of failure that I can fix? I'm multitasking because I have something that I want to show you that's kind of an answer to your question, but I need to find it first. I think it's in here. Yeah, here. So this is some data for a challenge that I participated in over the last couple of months on expansion microscopy data, registering expansion microscopy data. And it included data from 3 different organisms. It had C. elegans, that's the top row here, zebrafish, middle row, and mouse, bottom row. Let me pause it there. You can tell from this speckle, the mouse data was pretty low quality data in a lot of ways. First, both the fixed and moving image for a lot of this mouse data had low signal to noise ratio to begin with. But second, and potentially more problematic, is that between the fixed and moving rounds, there was a lot of signal dropout. So in the moving round for these mouse samples, the features present in the image were a little bit different than what was present in the fixed image. And the nature of the intensity in the image, how objects appeared, was also quite a bit different. Just looking at the images, I was pretty skeptical about being able to register them, but the point of this video is to show that I actually was able to successfully align them. And what made it work in this case was starting with a really simple transform. If you have really tricky data and you start with like a complicated deformable alignment, it's bound to fail. If you start with something really simple, like I just want rigid alignment, all I want to do is translate and rotate the data, you can probably succeed. You need very, very few features to successfully rigidly align two data sets. And then once you have a really good rigid alignment, now it's easy to get an affine. And now once you have a good affine, I'm not going to say it's easy to get a deformation, but it's easier to get a deformation. So my first answer regarding automated registration tools is that even when you have low quality data, if you strategize how you do the alignment in a way that is like really gentle and careful, you can still succeed. And then my second answer is that humans are always better at computers at recognizing what two things should go together. So a manual tool is a really good way to start. If you start with big warp just to get the two objects right on top of each other, or like get out some of the like really tricky to deal with distortion, then an automated might work very well after that. Conversely, if you do a nice, gentle, rigid automated alignment that works well, maybe a manual alignment after that is really less time consuming because the correct corresponding objects are much more apparent when you're looking at the two images. So there's no, hard threshold for registerable versus non-registerable. Every data set's unique, and tricky data sets just require different strategies.
Is there a reference that you would say that would help somebody starting out to determine what strategy to use, like a decision tree like strategy chart that would say like, do this at first or try this?
Sure. Yeah. I mean, not one written down, but I can make a note to start to like produce something like that. That might be really a good idea. But
and is this the Jupyter Notebook and data? Is it available like on GitHub or something like that? We can.
I'll put this on the how-to wiki page, the slides and the Jupyter Notebook. The Atlas data, I can 100% include in that. I'll have to ask my project team if I can include this specific zebrafish image, but I don't think it'll be a problem if we share that with JLANs.
And the link to wiki is also in the Hughes Hub calendar invitations.
I ask like a rhetorical question and then another question.
Please, yeah.
Can you talk about how or why it's important to store the transformations and not just the image that comes out?
Oh yeah, because that's the two registered images themselves. Like I still have this open, right? So this is and I still have it stuck behind the Zoom thing. There we go. All right. So if you have two images that are registered together like this, it is impossible to recover the transform that registered the two data sets. Like you can look at them and say, yes, those images look nicely registered. But if you want to use that registration result to do any of the other things I discussed, like moving objects back and forth between the two spaces, or even just study the transform itself in order to learn how the shapes of the two different objects were different, that's all contained in the transform, not in the image data itself. The image data, once you've resampled it like this, the only reason to do that really is to visually confirm that you have found a good transform. But the important object is certainly the transform itself. One way to just see quickly that it's non-recoverable is that the image data is scalar valued. At every voxel, you just have a single number. The deform data is vector valued. At every voxel, you actually have a displacement vector. So there's not enough information in the resampled image to reconstruct the much higher dimensional object, the deform. So that's a common, I think, thing that very new registration users will do because they're very focused on the image. I want to see that things are aligned. And once they see it's aligned, then they feel job done. But remember, it's the transform object that created this correspondence, and it's what contains all of the work you did in the registration algorithm is in the transform object itself. That's the very important thing to store.
Awesome. And when you're tweaking parameters, I guess, could you talk just a few minutes about, like, in your experience and for the data you work with, like, what are the parameters that you find you're tweaking the most? And
Sure.
I guess what decisions go into which parameters you end up tweaking?
Sure. This is the art and the special sauce of registration. And I compare, and it dovetails it unites very nicely with the previous question about like a flow chart or like a good, tools to help people learn to become better at using registration tools, not just how to use it, but how to use it well, how to wield it well. it's kind of like preparing samples at the bench, right? There's a lot of like someone can teach you these are the steps, but without a lot of practice, you're going to destroy a lot of samples first. And there's also a lot of voodoo in it where it's like, well, if you do it during a full moon, it works, but if you don't, it fails. So because these things are complicated, you know, it takes a lot of tinkering sometimes to get it right. And that's one reason why I leave a lot of these parameters exposed, is to encourage the user to read about them. That's the first thing I should probably say, is that at least for BigStream, all of these parameter values are documented somewhere. At the place where they are used, there is a long doc string explaining what that parameter does, what impact it has on the algorithm. So it might take a little bit of digging, but you can find it yourself in the documentation and start thinking about, oh, well, my alignment seems to have failed because the object or the image wasn't allowed to move enough. What parameters would give the image more freedom to move more quickly? Or it might be my alignment failed because it took 4 days and I just can't wait that long to see results. I need it to be faster. So which parameters can I change to make things faster? All of those types of questions can be answered by reading the doc string and putting some thought into it. But then to share with some of my experience, the very first thing I usually change is this smooth sigmas parameter. What this does is it applies a Gaussian smoothing to both the fixed and moving images before you start registering them. One of the most common ways that registrations can fail is that they get stuck into local minimums. Maybe you have like the top half of the two images are aligned really well, but the bottom half of the two images is not aligned very well. And if you were to wiggle the moving image, you might be moving the bottom halves into a better arrangement, but moving the top halves into a worse arrangement. And when you're doing that, you're leaving that local minimum, and the algorithm hates that. It wants to stay there once it finds it. And by smoothing the images a lot, you're eliminating a lot of those local minimums from the search space and preventing the algorithm from getting stuck. The second thing that I play with a lot in a related way are these shrink factors and alignment spacing. In different ways, these two numbers control at what resolution the alignment is actually done. So this fixed image, for example, has 210 Z slices, 1,025 X pixels, and 1,945 Y pixels. That's a lot of pixels overall, volumetrically. We don't need all of them in order to align accurately. We can downsample a lot. And these two parameters in different ways control how that downsampling happens. Often it's better to make these big values and throw out more data, especially early. Usually you want your first few steps in alignment to be really simple, like just put the two shapes on top of each other. And once you feel confident you're doing that, then you gradually refine. The real guiding principle for successful automated alignment is coarse to fine, both in image resolution and in image features. And these downsampling parameters give you coarse resolution, and the smoothing gives you coarse features. It blurs out like little noise patterns or small objects that might trip the algorithm up. So, and then finally, these optimizer arcs. So these are the parameters that I leave exposed explicitly in a lot of my example notebooks because they are the ones I change the most frequently. But there are like another 40 that you can change. Don't let that intimidate you. Really, it's just more power and flexibility. In many cases, you will never have to touch them. It's good that they're available to change because you have more control over the algorithm, but it can be overwhelming if all of them are in front of you all at the same time. These are the most common ones. And then these optimizer arguments, they control the gradient descent part. So I also very frequently am fiddling with this minimum step size argument, which is basically the convergence criteria. The larger this number, the earlier the algorithm will converge. And at a 0, it's just going to run and run and run and run and run until it hits the number of iterations. So I tweak that a lot too, to compromise between speed and accuracy. If I notice that like I'm spending an extra 20 minutes at the end of the algorithm to move one voxel, really just remove that part from the alignment.
That was fantastic. Thank you.
So Greg, I have one more question. So let's give a scenario here, like you have an atlas, you have ROIs in that atlas. And then you have a series of experiments over time where you have a 3D object over time. Would you take the ROIs and move them to the object, like your the time series data, or would you move the time series data, transform that, and then take the same ROIs within the Atlas space to that? Like, what you go either
Plenty
way.
of-
I guess you can produce artifacts depending.
Mhm. Plenty of strategies there, and I think it depends on which alignments are the easiest. So if like doing a tracking of your moving objects in your subject space is possible and easy to do, or more easy than doing a ton of atlas to individual time point alignments, then that's what I would do. I would do some sort of tracking or registration from time point to time point of my time series data, and then only once register the atlas to the first time point. It doesn't even have to be the first, to whichever time point is the best, the easiest to register to. And then now you have everything, you have a transform connecting all the things you care about. You can move the labels onto one of your time point subject data. And then because you have tracking, you can move those labels within the time, along the time axis between different acquisitions. That to me seems, if it can be done, is my preferred strategy. And we do that with our zebrafish data for calcium imaging. We usually do motion correction and stabilization in the time domain for the functional imaging data, and then register only one time point to the atlas. Mhm.
Greg, how do you usually keep track of what parameters you use to execute a specific pipeline?
Tons of random Jupyter notebooks stored everywhere. Non-rigorously is the answer. It's all up here in the old bean. There's no like principled way of doing it. It would be kind of cool if there was like a repo like mechanism for like storing parameter sets and doing grid searches and stuff. That would be great features to have, but I haven't ever taken the time to implement them or look for them.
Okay, so it sounds like your current system is you have a specific build book or specific data set or pipeline.
Yeah, I think of it more like I sit down in front of you know what a mixing board looks like for like audio mixing? It's got tons of knobs all over. That's how I kind of think of my tools, is like I sit down to do a registration experiment. I'm like sitting down at one of those mixing boards, and then I load the data up. I run a quick and dirty alignment, and then I start turning knobs. And then usually within a little bit of tinkering, I get a result I'm happy with, and that's it. I just store the notebook and say, these were the parameters that ended up working. I think of them more as like live adjustment knobs than like, here's the set that's always going to work for this data type, because that's not always true. I mean, these are all three C. elegans, these are all three zebrafish, these are all three mouse. In most of these cases, it's a slightly different parameter set that actually succeeds. Which makes automating large scale projects harder, because you have to go in and sometimes tweak things on your own for different subjects. But yeah, that's what's available, really.
I think that might be a good place to call it. So let's thank Greg one more time for a really awesome presentation.
Thanks, guys. One more plug. I'm giving 2 more talks after the how-to summer break on registration-related topics. They have considerable overlap with what I showed here, but with different specific focuses. I think one of them is on in vivo to ex vivo registration, which, similar to Atlas registration, you're doing a lot of the same stuff, but it's much harder usually because the 2 samples are really different. And so I'll try to focus on those particular aspects of in vivo to ex vivo. And then I can't remember what the other one is. Or it's like, well, but there's another one similar. It's like same kind of registration, but a specific focus on like one flavor of registration and what are the specific considerations for that. So if those are more in line with experiments you're doing or something you're interested in, keep an eye out for them, like September, I think.